To understand how organisms pass their traits along to their offspring, we're going to use the example organism of a pea plant because this is what a famous monk did in about the early 1800s, and his name is Gregor Mendel. Pea plants are very simple organisms. They have 7 pairs of chromosomes, and a lot of their distinctive phenotypes that you can see with your eyes are dominant. They're dominant or recessive. They're either yes or no. They don't blend. So they're very easy to look at and do an experiment on. And here are some of the traits. So we have the round, yellow, white, the color, the pod phenotypes, and then the stem. So this monk named Gregor Mendel was doing a small experiment. He used to teach a little bit, and he started doing these small experiments on pea plants, and it all stemmed from this observation he made. It was really important. So what he noticed was there were tall plants. That's a plant. I'll use a green. And there were short plants. These are pea plants, and you can see what they look like. And when you cross them, people at the time thought you would get medium plants. So I'll say tall, short, and here you get this blend of medium height. Okay, but that's not what he saw, and he was gardening quite a bit. What he saw was you had a tall plant and you had a short plant. You cross them, and what do you get? You get 100% tall plants. So tall, short, and this is tall. And then what happens? See, plants you can cross-pollinate with themselves. So he took two of these plants. He took 2 of them, and he cross-pollinated them, and he expected to get all medium. 2, 3, 4, all medium. So this is what the model was at the time. So what would happen if you crossbreed all the talls together? So if you took these 2 of these talls and you pollinated them, what would happen? Well, you should get all tall, right? But what he got was 3 tall. And 1 short. And it's from this observation and a lot of statistical observation that he made, because he didn't just do this with 1 or 2 plants, he did this with hundreds, maybe even thousands of plants, and he started seeing the numbers, and it was always 3 to 1. 75% tall, 25% short. So let's take a closer look at the pea plant. The pea plant has 7 chromosomes, 1 from the mother, 1 from the father, 1 from the mother, 1 from the father, mother, father. So this is 1 pair, 2 pair, 3 pair, 4, 5, 6, 7. And we're just going to look at the height, which is on the first chromosome. So T for tall, little t for short. Remember, chromosome is a coiled DNA holding the genes. It has many genes, but we're just looking at the one for height. And we're going to lose the chromosome. We're just going to use the letters now. So he had one plant that was capital T, capital T. That was this one. This is capital T, capital T. And he had one plant that was short. Little t, this one. And he bred them. Now, in this situation, the chromosome can come from either one. Either one of these chromosomes can independently assort themselves, and you can get either one, okay, because they segregate. So let's just say the next plant gets this one. You only get one from each parent. So this one. and let's say you get this one from this parent. Now we have a plant that is heterozygous. So this is homozygous dominant, homozygous recessive, heterozygous. But is it tall or short? So this is tall, short, and this would be tall. This would explain what Mendel saw. There is no medium. He never saw a medium. Now, if you took two of these, let's take two of these. This is much more complicated because there's more options. And you bred them together, you would actually get 3-to-1 ratio. But how do we do that? We do that with what is called a Punnett square. So let's take a closer look at this cross. And the way we do that is we draw a Punnett square. And this will be our first example of a Punnett square. This is going to give us the statistical ratios of each child, the chance of each child. What we do is we put these genes and we segregate them completely, so they're no longer really related to each other or touching. This is going to be from, let's say, the father, and this one up here is going to be from the mother. So these go here, and they're completely independent. If they're not affecting each other at all, they'll segregate completely. So we're actually going to drop down. Let me change the color. So I'm going to make this actually red. So these T's right here will drop first, and you just distribute them across down through each square. So I put this one here, and I put another one here. I put here, and I put here. When we go across, we're going to do the same thing from the father's genes. Oops. Okay, so now if the big T is present at all, you have a tall organism, plant. If you don't have any big T, you just have two little t's, you will have a short plant. So let's take a look and let's label them. So this one right here has two big T's, capital T, so it's tall. This one has a capital T and a lowercase t, so the capital T would be dominant over the lowercase, so it would be tall. And this one has a capital T and a lowercase t, so this would be tall as well. This one right here has two lowercase t's, so it would be short. I'm going to circle the tall ones. 1, 2, 3. And take a look. We have three tall ones right here. Three tall plants. T, 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 little t, lowercase n. So this right here explained Mendel's observations. And at the time, they didn't really even know about genetics. They had not discovered DNA. So he did all this just with observation, and he made up three laws. Okay, so this is Mendel right here, and this is some of his notes. He was very statistical and mathematical, and he was able to see trends, and that's why he started making these observations that he said doesn't make sense with the current theory. He came up with these three laws, and I wanted to explain them just a little bit. So the law of dominance we're going to be hearing about quite a bit, because we're going to hear the words dominance and recessive. And that just means that basically one gene, one version of the gene can be dominant over the other, meaning it will be expressed if it's present. So if you have a big T and you have a smaller lowercase t, the uppercase T will be expressed. If you have the recessive will only be expressed if there is not a dominant gene. So what this means is there's no blending in the pea plants, at least when he looked at them. There are much more complexity in genetics today. But classical genetics, dominance and recessive, it's either a yes or a no, and you're only going to get one trait. Now the law of segregation means that the offspring is going to get only one of the chromosomes from the mother and from the father. So when they make an offspring, the father will only be giving it one, and the mother only one. So these chromosomes will be segregated from each other. Now the law of independent assortment means they'll be separated randomly. The child will receive the chromosome randomly. There's nothing that really they don't get influenced by the other chromosome. And if they are completely random, like a coin flip, you'll get pure statistical analysis.